Welcome to the Deep Dive. We sift through complex information to pull out the key insights, basically giving you a shortcut to being genuinely well-informed. Today, we're plunging into a really fascinating new study. It connects something incredibly personal, your gut bacteria, to health outcomes in a way you might not expect, specifically its role in cancer treatment.
Yeah, we're looking closely at excerpts from a brand new article. It's called Gut Microbiota in Advanced NSCLC Receiving Chemoimmunotherapy, an ancillary biomarker study from the Phase 3 trial JSOG 2007 NIPPM. It just came out in the Journal of Thoracic Oncology this March. Really cutting-edge stuff, and it has some, well, potentially big implications.
Right. And our mission today is to unpack how these trillions of tiny organisms living inside us, might actually hold clues about how well certain advanced lung cancer treatments work, and even maybe predict side effects. So for anyone curious about where personalized medicine is heading and how unique your own body chemistry is, this is where it gets truly interesting. Okay, let's unpack this a bit. We know immunotherapy has been a huge deal for advanced non-small cell lung cancer, NSCLC, but a core challenge is still there.
Yeah.
How do doctors pick the best possible therapy for each specific patient? Current tools, like PD-L1 expression.
Yeah, the standard biomarker.
Exactly. It helps predict response sometimes, but often it has, limited usefulness when you're talking about these newer combination therapies.
That's precisely it. And that's where the gut microbiota might just step in as a potential game changer. This study we're looking at, it was part of a big, multi-center, randomized Phase 3 trial, JCOG 2007, or NIPP1. They studied 270 patients, all with advanced NSCLC, and they were getting one of two different first-line chemoimmunotherapy combinations. One was pembrolizumab plus platinum doublet chemo, they call that PC. The other was nivolumab and ipilimumab plus platinum doublet chemo, that's NIC. And crucially, they analyzed fecal samples taken right at the start, baseline samples, to see if the bacteria there offered any clues about how things would go.
Okay, so they get the samples, they get the two treatment groups. What did they actually find? What was the connection between the gut and how well the treatments worked?
Well, this is where it gets really fascinating. The sheer variety of bacteria within one person's gut, what scientists call alpha diversity, that actually didn't predict overall survival.
Oh, okay. So it wasn't just about having lots of different types.
Not directly for survival, no. But the overall structure of that microbial community, the beta diversity, that showed clear differences. They saw distinct patterns between patients who lived longer, say over 12 or 18 months, and those who didn't. It strongly suggests it's not just how many types of bacteria you have, but really which specific types are dominant and how they're interacting in that whole ecosystem.
Right, like a specific team lineup matters more than just the number of players. So do they identify, microbial good guys in this lineup, bacteria linked to better outcomes?
They absolutely did. The study pinpointed several bacterial genera tied to longer overall survival. You can think of them as potential allies. Names like Fusicatenobacter, Butyrococcus, Blautia, Bifidobacterium, and also the Eubacterium ventriosum group. Having more of these was linked to a reduced risk of mortality, and that held up even when they accounted for other factors like the cancer stage, patient age, smoking history.
So more of those good bacteria, potentially a better prognosis.
Right.
Makes sense. What about the flip side? Were there bad guys associated with worse outcomes?
Yes, conversely, they found certain genera that were associated with a higher risk of mortality, a less favorable outlook. These included groups like Ruminococcata UBA 1819, the Prevotolacea NK3B31 group, Acinetobacter, and Lactobacillus. And this really raises a critical question, doesn't it? Could analyzing a patient's gut profile genuinely add to, or maybe even improve upon, existing biomarkers like PD-L1? Could it give a clearer picture of who's likely to respond well?
Yeah, moving beyond just treatment effectiveness. Cancer therapies often bring pretty serious side effects. Does our gut microbiota have a say in that part of the experience too?
It certainly seems to, and this study gives us some really valuable clues there. They found that patients who started with lower overall microbial alpha diversity, so less variety in their gut bacteria, they had a 2.6 times higher risk of experiencing serious side effects, grade 4 or higher adverse events.
Wow, 2.6 times. That's a pretty significant link. So a less diverse gut might mean a rockier road through treatment itself.
Potentially, yes. A much tougher journey. And, it wasn't just about diversity. While some bacteria like Actinomyces and Anaerococcus were more abundant in patients who did have serious side effects. What's really interesting is that many of those same good guy bacteria we linked to survival Blautia, Fusicatenobacter, Butyrococcus, Eubacterium ventriosum group, Roseburia, and others like Parasitarella, Lecnospora, and Stipes. These were often found in patients who didn't have serious side effects.
Ah, okay. So the good guys might be protective against side effects too.
Exactly. In fact, having a low abundance of these potentially protective genera was strongly linked to a higher chance of serious adverse events. Just as one example, having low levels of Fusicatenibacter was associated with a threefold higher risk of those serious events.
So the gut profile might predict not just will this work, but also how tough will this be. That can be incredibly helpful for managing patient expectations and maybe even intervening. Huge potential benefit. Now, you mentioned this study compared two different chemoimmunotherapy regimens, the PC and the NIC arms. Did the gut's influence differ depending on which specific treatment a patient got?
Yes, and this is where it gets really interesting for potentially personalizing treatment down the line. They found clear regimen-specific effects. For instance, those beneficial bacteria we talked about, Fusicatenibacter and Beta-Rhizococcus, they seem to offer a greater protective effect, so better overall survival, specifically in the patients receiving the NIC regimen compared to those on the PC regimen.
Interesting. So if you're on that NIC combo, having those particular bacteria might be even more critical for a good outcome.
Precisely. And then looking at the other side, that Previtalaceae NK3B31 group, which was generally linked to higher mortality risk. Its negative impact was particularly strong, specifically in the NIC arm. The hazard ratio was 2.33. It makes it look like a potential negative biomarker just for that NIC regimen.
Okay, so that's efficacy. What about side effects, regimen specifically?
Also different, lower gut alpha diversity, less variety, was much more strongly linked to a higher risk of serious side effects in the NIC arm. The odds ratio was 3.8, nearly 4 times the risk.
Wow, compared to the PC arm.
Yeah, in the PC arm, the odds ratio was lower, about 1.7, still a link, but much weaker. And furthermore, those protective bacteria, Fusicatenibacter, Beta-Rhizococcus, Blautia, when they were found in low abundance, that was associated with a higher risk of serious adverse events, specifically in the NIC arm. It really suggests that for the NIC regimen in particular, the gut microbiota might play an even more critical role in determining both how well it works and how well patients tolerate it.
This really paints a picture, doesn't it? Knowing a patient's gut profile could potentially help doctors choose not just whether to use chemoimmunotherapy, but which specific combination might be the best fit for their individual microbial landscape. That feels like the ultimate goal of personalized medicine, right?
It absolutely does. And if we try to connect this to the bigger biological picture, well, the exact ways these bacteria influence things are still being worked out. But there's definitely growing evidence that specific types, especially these butyrate-producing bacteria like Beta-Rhizococcus, Fusicatenibacter, and the Eubacterium ventriosum group, they seem to help enhance the body's own anti-tumor immune response. They might do this by, say, boosting certain immune cells or inhibiting things that suppress the immune system. This study really adds strong support to that idea.
It all sounds incredibly promising. But, with any study, there are always considerations. What were some of the maybe limitations or caveats here, things researchers need to keep in mind?
That's a really important point, especially thinking about applying this clinically. It is the largest prospective study of its kind for first-line chemo-IO in advanced NSCLC, which is great. But there are limitations. For example, they used 16S ribosomal RNA sequencing to identify the bacteria. That's a standard method, but it gives you less detail, less resolution than some newer, though often more expensive, techniques like shotgun metagenomics.
Okay, so maybe missing some finer details.
Potentially. Also, when they looked at the regimen-specific effects, the subgroups were naturally smaller. So those findings, while intriguing, probably need confirmation in larger groups. And a really key point. They only collected the fecal samples right at the beginning, at baseline. We don't know from this study how the gut microbiota might change during the treatment course itself.
Right, so showing a strong association at the start, but not necessarily proving direct cause and effect throughout treatment.
Exactly. It's an association, a very strong one, but not yet proven causation.
So clearly more research needed. What do you see as the next steps for this whole field? Where does it go from here?
Well, future studies definitely need to confirm these findings, ideally in larger, maybe more diverse patient populations. They also need to dig deeper into those underlying mechanisms. How exactly are these bacteria interacting with the immune system and the treatments?
Yeah.
It might involve looking at metabolites in feces or linking gut changes to markers in the blood. And then crucially, the next big step is exploring interventions. Could things like fecal microbiota transplantation or specific targeted dietary changes or maybe advanced probiotics or prebiotics Could these actually modify a patient's gut microbiota in a beneficial way?
Ah, so actively changing the gut profile.
Potentially, yes. To try and improve treatment response or reduce those definite side effects, if that proves possible, it could fundamentally change how we approach preparing patients for and supporting them through cancer therapy.
So wrapping this up, what's the key takeaway for you, the listener, the curious mind tuning in today?
I think it means we're genuinely on the verge of a pretty significant shift in how we think about cancer treatment. The gut microbiota, which was, largely ignored for a long time in this context, it's really emerging as a potentially powerful patient-specific factor, a biomarker, really. One that might help guide treatment choices and maybe even help manage side effects, especially for these complex combination therapies used in advanced lung cancer.
It's just a really compelling example, isn't it? How understanding these intricate ecosystems inside our own bodies can unlock completely new avenues in medicine. It pushes us closer to that goal of truly personalized care. We really hope this deep dive has given you some surprising insights and maybe a clearer view of this exciting research area. Thanks so much for joining us.
And maybe just a final thought to leave you with. If these trillions of microbes living inside us can have such a profound impact on how our bodies respond to life-saving treatments, what other hidden influences might our complex internal networks have on our health, our well-being, that we're only just starting to understand?